purpose of my circuit is to create a type of memory for a population of cells. In this circuit, an on signal will stimulate a continuous behavior in the cell, and the cell will communicate this signal to the rest of the population in order to turn the entire population on. An off signal delivered to the entire population will turn off the circuit. It is very similar to the toggle switch we previously studied, and the switch element is very much based on that design. The idea behind my circuit is to test the logic behind population memory based on a single cell's experience. I'm modeling this logic in prokaryotes, but if this logic can be implemented in humans, then we could potentially use it to produce artificial immune responses. For example, if the on input is a pathogen, the signal can propagate a response throughout the population to protect the rest of the cells from the pathogen. Then, once the pathogen is eliminated, an off signal can be delivered through intravenous infusion to turn off the response in all cells. So I started designing my circuit based on three principles. Firstly, the system starts off, and then an on signal, no matter how brief, turns on the circuit, and the circuit remains on until the off signal is input. So I decided to construct a logic gate representation of my circuit, starting with an on input and then an off input. The on input is linked to an OR gate, and the output of that OR gate is going to be the output of the system. The off input is linked to a NOT gate, and that NOT gate is linked to an AND gate. The AND gate is going to output to the second input of the OR gate, and the output of the entire system is going to be the second input to that AND gate. So let's look at our first state, where both the on input and the output are 0. In this case, the output is 0 as well, and the whole circuit is off. But if we change the on input to 1, then because of that OR gate, the output is 1, since you only need one on input for the OR gate to have an on output. The output travels to the AND gate. Since the NOT off input is equal to 1 and the output is equal to 1, that AND gate is equal to 1, which inputs into the OR gate, making both inputs to that OR gate 1, and the output is still 1. Now if we turn off the on input, then the output is still 1 because of this OR gate. But if we turn on the off input, then that AND gate switches off, since one of its inputs is now 0. And so it doesn't support the system anymore. Because of that, both inputs to the OR gate are 0, and then the output becomes 0 as well, recalibrating the entire system to be used again in case it sensed another antigen. So going from there, how do we translate this into a genetic circuit? Well, I had to start off by choosing biological parts. I found a lot of inspiration for this from the toggle switch, which I'll talk about a little bit later. First, I decided that I needed the same two genetic codes that started off the toggle switch. One of these has a TET operon, and the other one has a LAC operon. The TET operon codes for a LAC inhibitor, and the LAC operon codes for a TET repressor. This way, the two circuits can repress each other. Then on the LAC operon, I also coded for a LUX-I and a GFP, which I will define as the output of my system. In order to have a continuous output in the cell and also have propagation to other cells in the same population, I'm going to be using a system with LUX-R, LUX promoter, and LUX-I. LUX-R is going to be constitutively produced by the left LUX promoter, and the right LUX promoter is producing the exact same thing as the LAC operon. This way, even if the LAC operon is turned off, we'll still have constant expression of the output unless a small chemical is put into the system to turn off the entire circuit. Here I'm just adding the repression of the TET-R to TET-O, and also the activation of LUX-R, which is bound by AHL, which is produced by LUX-I, in order to activate the right LUX promoter. IPTG is going to be our on input, which inhibits LAC-I, and ATC is going to be our off input, mixed with AHL analog, to be able to not only turn off TET-R, but also to occupy LUX-R, so the entire circuit output is turned off. In order to analyze the circuit, I'm going to be using MATLAB and GROW. I'll be using MATLAB for the temporal analysis part. For this, I want to look at the sensor cell and its reaction to the on pulse and the off infusion. With GROW, I'm going to be looking at spatial analysis, where I'm going to be looking at the signal propagation and how the entire population reacts to an off infusion. We make several assumptions in our model. We assume that the repressor as well as small molecule inhibitor reactions are fast, so we get the Hill equations. We assume that transcription and translation rates are the same for all proteins. Spatial analysis discounts GFP production from LACO and relies on GROW examples. We have many constant assumptions as well. Spatial analysis ignores the LUX-R-AHL dimerization, and spatial analysis also assumes that the HL analog has the same affinity for LUX-R as AHL does. Let's begin with the temporal analysis of the single cell. Initially, the cell is turned off, and then, at time 50, a pulse of IPTG is sent through the cell, a pulse of concentration 50 nanomolar. This pulse is then turned off about 100 seconds later. While the cell is in the off state, IPTG, TET-R, AHL, and GFP are low. LAC-I, on the other hand, is high, as it's inhibiting the output of all these proteins. Once the pulse of IPTG is put into the system, the concentration of LAC-I drops from about 40 nanomolar to about 12 nanomolar. GFP rises from 17 nanomolar to about 33 nanomolar. TET-R rises from about 14 nanomolar to about 27 nanomolar, and AHL rises from about 12 nanomolar to about 23 nanomolar. This makes sense of the circuit. However, the fact that GFP is greater than AHL here contradicts a little bit what we see in the spatial analysis. However, this may be due to the fact that we do not look at the IPTG pulse in the spatial analysis. Once the IPTG pulse is turned off, we see that the circuit still remains on with a relatively high level of GFP. However, it does drop a little bit along with TET-R and HL, and there is a slight rise in LAC-I. This might be due to the fact that IPTG may be binding more strongly to LAC-I than TET-R is to the TET operon. Perhaps, however, this would change if we took the diffusion of IPTG into account. When we add the ATC off input to the circuit after it's been turned on for a while, we see that it acts as expected. TET-R, which is around 60 nanomolar, GFP, which is around 31 nanomolar, and AHL, which is around 20 nanomolar, all drop to about 0 after AHL analog and ATC are added to the system. You can tell that the AHL analog is added a little bit later and also has a substantial effect, just as the ATC does. LAC-I levels rise from about 0 nanomolar to about 100 nanomolar after the signals are added. One thing to note is that the signals are at a very high level, each at about 1000 nanomolar once the signals are added. I tried with a few other concentrations, but it seemed only the high concentrations were effective. This makes sense and fits with my idea that you would need an infusion of a very large dose to be able to have an effect and turn off the on signal. Now let's look at the spatial analysis of the population of cells. Cells grow in an off state until the sensing cell releases an AHL signal, which propagates to the rest of the population. Then the cells start to produce GFP, and you can see this by the change in green color, and the purple is AHL. Then, at about 26 minutes, the infusion of ATC and AHL analog are added. As you can see, the cells are then depleted of their GFP. However, a few cells are still remained on, and remain so without any changes until the end of the program run. This supports my idea that a long-term infusion at a high concentration is necessary to turn off all the cells. Looking into the future, even though my model performed pretty well, there are some things that I would want to change or add. Firstly, I would want to add diffusion elements to the temporal model. I would also want to model cells that are not the sensor cells, but any cell receiving an AHL signal. And I would want to more confidently model in GROW. I use a lot of examples in GROW to be able to build my model, but I would want to be able to understand the language and actually use it wisely. I also, and finally, want to test this logic in eukaryotes, which would be the ultimate goal, because only in eukaryotes could we actually get this artificial immune response, which is the end goal.